Hi, let's think about the fungi. So, fungi, in class, I don't like to define what they are because their definition is, it's either phylogenetic or complex on a biochemical level. So the phylogenetic definition is just that they are a group. They are a phylogenetic group. They share a common ancestor that nothing else shares with them. And so they have this shared evolutionary history. But then also, their cells tend to work in a certain way. They don't have the metabolic diversity of bacteria and archaea. They are chemoorganoheterotrophs like us. They tend to excrete digestive enzymes into the environment that break down macromolecules like carbohydrates, and then the cells absorb whatever simple sugars or amino acids have been liberated by those digestive enzymes. And some of these are really spectacular, like the white rot fungus that can grow on wood can break down chemicals in wood that nothing else can break down. But they still don't grow into the wood. They mostly are outside the cells, and they excrete enzymes that break stuff down, and then they absorb whatever they can that's left over. Okay, what else about them? Well, they're eukaryotes like you and me. Their cells have all that silly eukaryotic nonsense like nuclei, mitochondria, membrane-bound organelles that bacteria and archaea don't have. And so their cells are also bigger than bacteria and archaea. And the other thing you'll always see about fungi is that they have cell walls made of chitin. Chitin is a particular carbohydrate with nitrogen in it. And it's so they have these kind of dense, tough cell walls. Chitin you'd also find in arthropods. Their exoskeletons are made of chitin. Okay, cool. So some fungi are macroscopic. You can see them in mushrooms or puffballs or whatever. Here is the big thing I want you to think about. So mushrooms are not like us. So what do I mean by that? What I mean is your cells are clones. They share identical DNA. Mushroom cells do not. They do not share identical DNA. So what do they have? Like, what's the deal with mushrooms? Why do I bring up their DNA? Mushrooms are reproductive structures where two different groups of clones have met in the environment. So remember, fungi reproduce asexually, and that's through things like budding or Hyphae, or sexually. And when they reproduce sexually, what's happening is a group that has been forming asexually, they've been reproducing asexually, so they're all clones of each other in a hypha. Two of those hyphae meet. And so they're two not perfectly related groups now want to mate. And so they together will build a mushroom that lets them mate and create like recombined descendants. So this is a really impressive thing they do. And so when you see a mushroom, what you see is a bunch of fungal cells that are getting ready to mate, and their descendants, the recombined scrambled up DNA, are in cells that line these gills. And those are going to be spores that are not identical to their parents. They're a combination of their parents. So that's what mushrooms are. They're where two different groups of fungi have met and they want to mate, and they want to make their spores float away in the air and colonize new environments. Okay, we'll come back to that. We will look at the life cycle of a mushroom in a little more detail in a bit. First, let's look again at yeasts and molds. Yeasts are single-celled. They make little colonies on petri dishes. So they don't form hyphae. They don't explore their world in an organized way like that. And then molds do make those filaments. They make the hyphae. And they will also make fungal spores that could be sexual or asexual. And again, fungal spores are not endospores. They are not those almost indestructible things that some bacteria can make. Instead, they are just a common reproductive structure that can float away in the wind. Some fungi can grow as molds or as yeasts. So one clear example of this is Candida albicans, which causes things like, thrush. So that's oral candidiasis and vaginal candidiasis, or yeast infections. It also can be part of our normal microbiota growing in our gut without causing any problems. And what we find is when it grows into our tissues, so when it's actually causing a disease and invading our tissues a little bit, causing a lot of irritation, it grows as a mold. These hyphae invade our tissues. These hyphae invade our tissues. When it's growing harmlessly, it's growing as a yeast. So don't believe that molds and yeasts are like different groups of fungi. That's not quite what it is. So yeasts can have a lot of different shapes, and in this case you're seeing budding yeast. So that means that a parent cell will just grow small daughter cells out of it. And it's still reproducing through mitosis. It's still making new nuclei to give to the daughter cells. It's just not splitting in half. It's not doing a binary fission kind of thing. Yeasts can grow, well again, they're chemoorganoheterotrophs like we are, but they can grow anaerobically if they have to in many cases. And yeah, so I think that's what I just said. And they can make these things that are like pseudo hyphae because they don't fully separate with their daughter cells. But they don't quite have the same thing as hyphae have, because hyphae can grow very long and they're very intentional, whereas pseudohyphae might not be so much. So hyphae are these complex, amazing things. They are filaments that explore new territory. So what they're made up of is cells where each of these things is a nucleus. And so they kind of, they just copy their nuclei and they expand through mitosis asexually to get longer and longer. And there are connections between the nuclei in that the cytoplasm can flow a little bit between them. So this nucleus can touch cytoplasm that will then flow to touch this nucleus. So they can share nutrients that way. And in hyphae that don't have any septa between them, anything can freely flow along them. And that allows them to share the nutrients they find. So as they explore and expand into a new environment, the older cells at the back can benefit from that. So hyphae have very high surface area. They're very good at exploring their environment. And again, they release the enzymes that break down chemicals, and then they can absorb the nutrients that are released. It's cool. And then a mycelium is a kind of an organized mass of hyphae expanding out from a single spot. Some things to think about with fungal growth, and I think I will end this video at this slide. It's getting kind of long. Water, again, is the most important thing. They need some of it. So some fungi can grow in really dry environments, but most of them grow best when there's some dampness. And I'll show you some pictures of that. And for food, they need simple sugars and amino acids they can absorb, but they can get those by releasing digestive enzymes that liberate those from other macromolecules in the environment. And some of them need oxygen, some don't. They don't tend to grow at extremes of temperatures. They're more like us, and they tend to like roughly neutral or slightly acidic pH. Okay, let's stop here, and we'll see each other at the next video.